Good morning, students. Can you hear me?
Yes, sir. Good morning, sir.
Good morning.
Happy Teachers Day, sir.
Oh, thank you. Thank you very much.
Happy Teachers Day, sir.
Happy Teachers Day, sir.
Teachers Day, sir.
Teachers
Day, sir.
Thank you. So since morning, we are very busy with saying all the Happy Teachers Day because all faculty has come up with sarees, ladies, and taking photos also. So nice to see. Okay, so today we will start with one experiment that is DNA as a genetic material. Then probably after that I will distribute a seminar topic for you for membrane biochemistry for first 7 students, and the rest of the 6 rest of the 6 students uh, Vidya madam will give topics. So basically, the seminar topic, unlike last time, this time you have to prepare 10 slides and you have to present live, like Zoom. Same what I am using Zoom, you can also create a Zoom link. Then we will have a presentation by sharing the screen. So it doesn't matter whether you make 3 slides, 4 slides, 10 slides, 20 slides, but you have to finish everything in 15 minutes. Okay. So that's the seminar topic I will give you once I finish the class. So basically, yesterday we have discussed this DNA as a genetic material, right, in bacterium and virus. So here you can see bacterium. So basically, this experiment was done by which scientist? Can anyone guess? He injected a rough and a smooth strains into the mouse, and his conclusion was DNA is the one that is transforming into the cells and making R cells into S cells. Can anyone tell which scientist has done this experiment?
Griffith, sir.
Griffith, Frederick Griffith. Nice. So basically, we have talked about the bacterium. The DNA in the bacterium will convert a virulent bacterium into a virulent bacterium by taking over of the whole cell. So this is a bacterium, right? So next, Hershey says they have used virus. I mean bacteriophage. So the definition of bacteriophage is nothing but a virus that infects the bacteria is called bacteriophage. So now basically we have discussed these two, bacterium and virus, how the genetic material, which is also called DNA, enters into the another cell and taking over of the genetic material of the cell and destroys and makes new particles. So this time I will explain the DNA genetic material in eukaryotic cells, like humans, like mouse, I mean whatever the multicellular organisms. All these are single cell organisms, right? So this 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 experiment scientists have done in petri dish. So what they have done is they have taken the dead cells, I mean dead eukaryotic cells. For example, just imagine they have taken the mouse cells. So mouse cells they have taken and they have knocked down a gene called thymidine kinase. So, I mean, there are a lot of techniques available nowadays, how to knock out the gene in the cells or mouse or, I mean, anywhere. So, knockout mice are available throughout the world where one particular gene is absent. So, that is done by recombinant technology. So, basically, here in this slide, the first petri dish, the blue particles, blue dots are cells that have a TK gene deficient. So basically, these blue dot cells does not have a thymidine kinase gene in its cell. So if a gene, thymidine kinase gene is not present in the cell, what does it mean? Can it make a thymidine kinase protein? Can anyone tell? If a gene is absent in the cell, particular gene, thymidine kinase, is absent in this cell. So, can this cell make thymidine kinase enzyme?
No, sir.
Yeah, because thymidine kinase gene will be converted into RNA, which is mRNA, and mRNA will go into the cytoplasm, sits on the ribosomes, and start translating into a protein. So basically, thymidine kinase gene will give thymidine kinase protein. And the protein is the one that ultimately go and roam in the cells and function. So basically, these blue dot cells doesn't have a thymidine kinase. And if thymidine kinase is not present in the cell, they will die because that enzyme is much important for its survival. So basically, first petri dish, you have a dead cells because they don't have a thymidine kinase. So what you do is you take the TK thymidine kinase DNA. I mean, it is easy. How you take the TK DNA gene is very simple. So you take some cells that have a TK thymidine kinase DNA, then you crush it, then you isolate DNA from the cells. So the DNA that you isolated from the cells will have huge number of genes, right? Whatever the genes that are present in the cell, you have collected the DNA by technique called isolation of DNA from the cells. So now we have to, for example, there are 1,000 genes present in your DNA. Now you have to single out the thymidine kinase gene from the 1,000 genes. What you do is you take the DNA, then you use a primers specifically that transcribe the thymidine kinase. So you design a primers such a way that it will start making replication of TK gene. So you take the sequence of 5' sequence, forward primer and reverse primer of thymidine kinase gene. Now you take the DNA, you add the primers, you add the DNTPs, and you add the DNA polymerase. So then you run the polymerase chain reaction, which is known as a PCR. So once you do the PCR, these primers will go and bind to the DNA of the thymidine kinase only, because you have designed the primer such a way that these sequences will go and bind to the gene. So once the primers will go and bind to the DNA, what will happen? There are steps, right, in PCR: denaturation, elongation, initiation, elongation, termination. right? So similarly, like a replication, this TK thymidine kinase DNA will make huge amount of DNA. Then what you do is you take the PCR product and you run the agarose gel. I think this semester you have practicals of PCR. You will know how PCR can be done and how you visualize the specific gene. So out of 1,000 genes, you have replicated only thymidine kinase gene by adding primers, DNTPs, and DNA polymerase. So now you take the PCR product and you run the gel, agarose gel. Agarose gel is for DNA and Western blot is for proteins. So once you run the gel, there must be some size of the gene, right? Some 600 base pairs or 800 base pairs or whatever it is. So at 800 base pairs, you will identify the thymidine kinase PCR band. You cut the PCR band where the gene is present, then you purify the thymidine kinase gene. So that's how the specific gene of your interest of DNA can be isolated from 1,000 genes. What technique you have to use? First, you have to extract the DNA from the cells that may contain huge number of genes. Second, immediately your gene of interest, you have to design the primers. So once you design the primers, you run the PCR. PCR will amplify only gene of your interest. Then you run the agarose gel. Then you cut the PCR band that is present in the agarose gel. Then you purify the gel, which will give you only DNA. So now you have a pure thymidine kinase DNA in your hand after several steps. Then you take it in the tube, like here, add thymidine kinase. Then you add this thymidine kinase DNA to the cells. So you wait for 1 or 2 days, 24 hours or 48 hours. Then immediately you start seeing some live cells. So what it does, the thymidine kinase will enter into the cell and start replicating in the cell, dead cells. So why these cells were dead? Because there is no thymidine kinase. So once this thymidine kinase is present in the dead cells and it requires for cell survival, immediately the cells start dividing and make new cells. That's how you see some live cells here. Still, for example, if you have taken 100 dead cells, not 100 dead cells will take this DNA. Probably some of them are very bad and some of them are good. So only the cells that are good, they will take the DNA and they start dividing. So once they start dividing, they divide this thymidine kinase gene also. That thymidine kinase gene will go to their progeny. So then it is a normal cell like TK thymidine kinase present cells. So what this experiment will tell you is, if you have a foreign DNA and if you insert it to the cell, then that DNA will go into the cell, then it start making protein of that specific gene. So which tells you that DNA can be transformed, and this transformation principle is the only one that gives you a DNA is a genetic material. Why? Because this DNA will go to their progeny. Like if I wanted to tell you very simply, I have a 23 pairs of chromosomes, right? 46. So my kid will have half of my chromosomes and half comes from mother. So basically he will again have 26 pairs of chromosomes. So once he had a kid, he will transmit his half pairs of 23 pairs of 23 chromosomes to his son, and remaining 23 comes from mother. So basically, the 46 chromosomes will match. Only thing is, the genetic material or the chromosomes or DNA will go into the progeny. That's why it is called a genetic material. So now, as I told you in the slide, the transcription, why this is a transcription, why not it is an infection? So infection means if a virus or if a bacteria attack you, then it is called infection, because virus will have a nucleus and DNA, and the nucleus is surrounded by a protein coat. If you take the bacteria, it will have a membrane also. So basically, the whole bacterium, which contains lot of parts of it, will come and infect you. That is called infection. So what is a transfection? So for example, you have a bacteria. What you do is you extract the DNA from the bacteria. Then you left out the proteins or cell membrane, whatever it is. If you extract the DNA, and if you are giving this DNA into the cells, that is called transfection. So in a simple word, if I can define that what is a transfection, insertion of foreign DNA into the cell is called transfection. So infection is a complete virus or bacteria entering into the cells is called infection. So that is the basic difference. So why this transfection is useful nowadays? For example, if somebody is lacking a gene called probably insulin. So one person think that one person is having mutation in the insulin gene. So he is not able to make a insulin protein. So now what we can do is we can extract the his sperm and oocyte. Then we can insert a normal insulin into the eggs. I mean the oocyte. So the normal insulin will go and bind to the genome so that it will start making a protein. So that's how this is still unethical nowadays, because so much research has to be done on the humans. It was only done in the animals and cells. So in future, probably after 20 or 40 years, we can detect immediately what genes are mutated in this person. So for their next generation, for his son or for his daughter, you can stop transmitting that mutant gene by correcting it at the stage of uh, fertilization. So it will take a lot of time. That is the main important of transfection. So it cannot be done so easily, because if you add cells and DNA, DNA cannot directly go into the cells. You have to give some pressure or some heat shock so that you force the DNA to enter into the cell. That is transfection protocol. I can give you, once you come for lab, you will have transfection also. So in a transfection, we have a 13-step protocol, what reagents you have to use and how you force the DNA to enter into the cell. Because the cell has to rupture, right? because the cells are surrounded by membrane. So it will not accept any DNA. It has to rupture. So we make rupture, we force the DNA into it. So that is a transfection protocol. But basic understanding of transfection is insertion of foreign DNA into a cell. So basically, if I can tell you, as I told you before, here you have a poliovirus. It's a round structure. The upper part is a protein capsule. Inside you have a genome. So once you add this poliovirus to the cells, that is called infection, then it will affect the cell. Probably if it is a danger, it will damage the cells. So then what you do is you break the whole proteins, you extract DNA, viral RNA, then you can directly add any, either it is viral RNA or viral DNA. So there is a process called, yesterday I told you, right, reverse transcription. If you have RNA, you use the reverse transcription, then you make a poliovirus complementary DNA. You can infect this one also, even you can infect RNA also. If you infect RNA, still the RNA has to undergo cDNA in the cells. So in order to avoid that, of course, you can add anything. Poliovirus DNA also can be added, cDNA. So this DNA will enter into the cell, then it starts making poliovirus particles. So that's the infection. That's the DNA is taking the control of the genome of the cultured cells. Then it starts making the poliovirus DNA. Because this poliovirus DNA will have all the protein capsules that are necessary for poliovirus, right? So that is called a transfection, and this can be used for the humans also in the near future. So basically, this is the 3 experiments that will tell you that DNA is a genetic material. One is a Frederick Griffith experiment, the mouse experiment. The second one is the Oswald Avery experiment, where she has done a very simple experiment by adding radioactively labeled sulfur and phosphorus. And the third one is Hershey-Chase, the bacteriophage, where the virus will insert its DNA into the bacterium cell, and the viral bacteria will damage the bacterial DNA and it start replicating like a bacteriophages. So basically from the here, you will see a lot of bacteriophages will be made. And the third one is the eukaryotic cells, where you can add a naked DNA or isolated DNA to the living cells and it start making new cells once it is active. And this process is called a transfection. So this is the one that gives you an overall idea of DNA is a genetic material. In some viruses, RNA can be acted as a genetic material, but those are very exceptions. Like in life, there are a lot of exceptions, right? In a similar way, sometimes some very few viruses will have a RNA as a genetic material. Next class, I will explain where RNA acts as a genetic material. So now I will give the topic. So who is the first one? First number? Hello.
Aishwarya, sir.
Aishwarya, she is here.
She is absent, sir.
She is absent. Okay. Can somebody write down for her semi-conservative replication? No,
Okay,
sorry. Sorry, it's a molecular biology. No. Okay. I'm talking about membrane biochemistry. So for Aishwarya, it is the physical properties of bilayers.
Okay, sir.
Who is next?
Sir, Alodi.
Alodi, for you, it is a lipid wraps.
Lipid?
Lipid wraps. RAFTS.
Okay,
Got
sir.
it? Okay. So who is next?
So me, sir, Ashwin.
Ashwin. Ashwin, for you, it is a Singer Nicholson model. Singer Nicholson fluid mosaic model. Got it?
Okay, sir. Thank you, sir. Yes, sir.
Who is next?
Divya, sir.
Divya. Okay. Divya, for you, it is a types of integral membrane proteins. So you got it?
Okay, sir. Thank you. Yes, sir.
Yeah. Who is next?
Sir, Padma Shri.
Padmashri. Padmashri, for you, it is a erythrocyte ghosts.
Sir,
Erythrocyte
erythrocytes.
ghosts. GHOSTS. Ghosts.
Ghosts, sir.
Yeah. Erythrocytes ghosts. You got it?
Yes, sir.
Okay. Who is next?
Punyashri, sir.
Punyashri, for you, it is a liposomes and micelles.
Okay, sir.
Okay, who is next? Who is after Punyashri?
Sir, you told only 7 students, no, sir?
Oh, 7 are finished. Huh?
I think yes, sir. 7 are done.
Okay, fine.
One more Pooja Shri should be there, sir. She is 7.
She's 7. I finished only 6 actually.
Yeah, Pooja Shri, sir, after Punyashri.
Yeah. Where is she? Is here? Hello?
She's here, but her mic is not proper, sir.
Okay, can somebody write down for her? It is ER retention signals.
Sir, repeat it, sir.
Endoplasmic reticulum. Endoplasmic reticulum. Retention signals. Retention.
Okay, sir.
So 7 people are done, right?
Yes, sir.
Okay, so I will inform Vidya madam also to give the remaining 6. So from next Saturday onwards, so Saturday you have a class, right? So every Saturday, probably I will finish 20 minutes of the class. So next 20 minutes you have to give your presentation. Okay. So it doesn't matter how many slides you prepare, and you can make a just same like a Zoom presentation, PPT presentation, and you present in the Zoom. Okay. So you got it, right? Hello.
Yes, sir.
Okay.
Sir, in one class, 2 members, sir.
No,
How many have to
only,
present in one class?
no, only one. One Saturday, one person.
Okay, sir. So from next Saturday it starts, sir.
Yeah, it starts from next Saturday, and
So next Saturday Aishwarya should do.
yes.
Okay, sir.
15 minutes she will present, and probably 5 minutes for questions. Okay. So this is the class. So I will finish now. Okay. Thank you all, and happy weekend.
Thank you, sir.